Here is a claim that flips a decade of longevity advice on its head. On August 4th, 2026, a study in Cell Metabolism found that the very diets we praise for extending life, the plant-heavy, protein-light Mediterranean and Okinawan patterns, can quietly leave you weaker, frailer, and less physically robust as you age. Now, to be clear from the outset, that frailty finding was seen in mice, and it has not yet been demonstrated in humans. So, hold the excitement with an open hand. And the fix the researchers landed on is not what you would expect. It is not cutting protein even further, it is adding back one specific amino acid, methionine, in a carefully measured amount. Get that dial right, and in mice, the same diet burned fat and protected muscle strength at the same time. Again, in mice not yet shown in people. So let us talk about what this protocol actually is, because it sits right at the center of one of the oldest debates in aging science. For years, two camps have argued past each other. One camp says, protein is precious. You need plenty of it to hold on to muscle and avoid becoming frail as you get older. The other camp points to the animal data, where restricting protein, and methionine in particular, is one of the most reliable ways to extend lifespan in yeast, worms, flies, and rodents. Both camps have real evidence. Both cannot be fully right. The 2026 study tries to resolve that tension. The researchers built a diet they call low-protein methionine-supplemented, a mouthful. So let us just call it the tuned diet. It models the low animal protein pattern of the Mediterranean and Okinawan diets, but instead of letting methionine fall as low as possible, they deliberately added a moderate amount back in. Why is this compelling and a little controversial? Because it suggests the longevity crowd may have been optimizing for the wrong number. The goal was never the lowest possible protein. The goal, this work argues, is the right amount of one amino acid to keep you both metabolically healthy and physically strong. To understand why methionine matters this much, think of it as a master signaling molecule, not just a building block. Your body reads methionine levels as information about how much protein is coming in. When methionine is scarce, cells switch into a repair and recycling mode. They ramp up autophagy, the process where cells clean out damaged parts and reuse them. In animal models, that autophagy-driven housekeeping is a big part of why methionine restriction extends lifespan. But here is the catch. Push methionine too low, and you also suppress the signals that maintain muscle and physical robustness. You get the metabolic benefits and the frailty at the same time in mice. That is the paradox. What the tuned diet did in these mice was reshape an entire hormonal cascade. 3 hormones went up: growth hormone, GLP-1, and FGF21. One went down: insulin-like growth factor 1, or IGF-1. That combination is unusual and interesting. IGF-1 is a growth-promoting hormone strongly linked to accelerated aging when it runs high, so lowering it is generally considered good for longevity. Growth hormone rising while IGF-1 falls is a specific signature that seems to preserve tissue maintenance without flooding the body with growth signals. Then there is FGF21. Think of FGF21 as a metabolic stress hormone that tells your body to burn fat and become more sensitive to insulin. It is the same pathway triggered by fasting and by protein restriction. GLP-1, you may recognize, it is the same hormone the blockbuster weight loss drugs mimic, involved in appetite and blood sugar control. The most important mechanistic finding is this: FGF21 was required. When the researchers removed FGF21 from the equation, the fat loss and the insulin sensitivity benefits of the tuned diet largely disappeared. So FGF21 is not just along for the ride, it is doing much of the heavy lifting. And separate research suggests FGF21 promotes metabolic health through channels that are distinct from its growth-suppressing effects, which is part of why this diet could burn fat while still protecting strength. So the picture is a diet that is low in protein overall, mostly plant-based, but tuned with enough methionine to keep growth hormone and the FGF21 system firing, in a way that protects the body while it remodels its metabolism. A picture drawn, so far, in mice. Now let us be careful about what was actually shown, and in whom, because this matters a lot. The core experiment was a mouse study. The researchers compared several diets head-to-head: a Western diet, a ketogenic diet, and the low-protein longevity diets. The Western and ketogenic diets both increased fat mass and frailty in these animals, and each brought its own metabolic cost, one pushing cholesterol up, the other worsening insulin resistance. The tuned low-protein diet with added methionine did the opposite. It reduced fat mass, reduced frailty, and improved cardiometabolic markers. That is a genuinely striking result because reducing fat and reducing frailty usually pull in opposite directions. They also tested a completely different approach as a comparison: bi-monthly cycles of a 4-day fasting-mimicking diet. That intermittent strategy independently improved metabolic markers, which suggests there is more than one road to some of these benefits. The part that reaches into humans is epidemiological. A cross-sectional analysis of data from over 200,000 men and women found that people with the highest animal protein intake had roughly double the prevalence of type 2 diabetes, compared with those eating the least. And this held even though the high-protein group tended to have healthier lifestyles overall. That is a large data set and a notable association. But here is the honest limit. The mechanism, the whole methionine tuning, FGF21 required story, comes from mice. The human data is observational and cross-sectional, meaning it captures a snapshot and shows correlation, not proof of cause. It cannot tell us that adjusting methionine will reduce frailty in people. A companion commentary published the same day by researchers at the National Institute on Aging framed this work as helping explain why plant-rich longevity diets sometimes compromise physical robustness, which is a useful hypothesis, but still a hypothesis in humans. And separately, decades of animal work and large meta-analyses do give us real anchors. Methionine restriction robustly extends lifespan across species, and Mediterranean diet adherence is consistently associated with lower all-cause mortality across large human cohorts. So, the surrounding evidence is strong even where this specific mechanism is still early. So what do you actually do with this? Let me be clear up front. The precise methionine dose used in this study was set for mice, and there is no validated human methionine target coming out of this work. Anyone who hands you an exact milligram number for people is going beyond what the science supports right now. What you can reasonably take from it is directional. The pattern that looked best was mostly plant-based and relatively low in total protein, but not methionine-starved. In practical food terms, methionine is concentrated in animal proteins, meat, fish, eggs, and dairy, and is generally lower in most plant proteins, with legumes being especially modest in it. So a diet built largely around vegetables, legumes, whole grains, nuts, and olive oil, with small, deliberate amounts of higher methionine foods like fish or eggs, is the closest real-world translation of what the tuned diet was doing. Before you make any significant change to your protein or methionine intake, though, sit down with a registered dietitian or your doctor and plan it with them, especially if you have any existing medical condition. A professional can look at your labs, your medications, and your actual eating pattern, and tell you whether an adjustment makes sense for you, rather than you guessing from a mouse study. If you already follow a strict plant-based or very low-protein longevity diet, and you have noticed you are losing strength, feeling weaker, or struggling to maintain muscle, do not treat that as a signal to start self-adjusting your diet. Treat it as a reason to raise the issue with your doctor or a registered dietitian, who can assess whether frailty is actually developing and whether your protein pattern is part of it. The takeaway is not to abandon the diet on your own. It is that going as low as possible on animal protein may not be the optimal move, and that is a conversation worth having with a professional. For tracking, the markers worth watching with your physician are the ones this study moved: fasting glucose and insulin sensitivity, your lipid panel, and body composition. Specifically fat mass versus lean mass, since holding on to lean mass is the whole point. Grip strength and simple physical function tests are cheap, practical proxies for frailty you can monitor over time. The fasting-mimicking approach is the other lever here, and some people find cyclical, short, low-calorie periods easier to sustain than permanently restructuring every meal. This information is for educational purposes only. Always consult your doctor before making any changes to your diet, supplements, or medication. And because this touches protein intake, blood sugar, and potentially medications, that conversation genuinely matters here. A few groups should be especially cautious. Older adults are already at elevated risk of sarcopenia, age-related muscle loss, and pushing protein too low without attention to strength could make frailty worse, not better, which is the exact trap this research highlights. Cutting protein is not automatically pro-longevity if it costs you your muscle. If you have diabetes or are on blood sugar-lowering medication, changes that improve insulin sensitivity can stack with your drugs and affect your levels. So this is a doctor conversation, not a solo experiment. The same goes for anyone on medication for cholesterol. Pregnant or breastfeeding women, anyone recovering from surgery or illness, and people with a history of eating disorders should not be restricting protein or running fasting-mimicking cycles without medical supervision. And remember, methionine supplements exist, but there is no evidence-based human protocol for them from this study. Self-dosing an amino acid based on mouse data is not something to do casually. What I find genuinely useful about this work is that it reframes the question. For a long time, the longevity conversation treated protein as a simple slider. Less is better, keep sliding down. This study suggests the truth is shaped more like a curve, where one amino acid needs to sit in a specific window, low enough to trigger the FGF21 and growth hormone signaling that burns fat and supports repair, high enough to keep you strong. In mice, at least, that window existed. Whether that window transfers cleanly to humans is the open question, and it is the thing to watch in the next few years of research. If you are running your own dietary experiment, ideally with a dietitian or doctor in the loop, I would love to hear which markers you actually track, the metabolic ones, the strength ones, or both, because that real-world data is where a lot of the interesting patterns show up.